So good afternoon, good morning actually, everyone, and thank you for joining us for Patterns in Plant Lives. This is the 10th in the Center for Environmental Inquiry's Dig into Nature series that we're doing this fall, a virtual events. We normally would have held these on the Galbraith Preserve in southern Mendocino County in Yorkville, or at the Fairfield Osborne Preserve on Sonoma Mountain in Renner Park. But because of COVID-19, we've gone to the virtual format, which we are finding offers a lot of different opportunities to bring in different kinds of people from different parts of the area to talk about different kinds of subjects than we might have otherwise. My name is Margot Rollins, and I am the program coordinator with the center. And I am just very happy to see you all here. And as I've already said, and I'm going to say again, if you're just entering now, if you would please put your name in the chat and the name of the town or area that you are from, so we can keep track of who actually attends the event. Let me tell you, before we get going on today's project, today's Phonology program, I want to just tell you a little bit about the center. The Center for Environmental Inquiry envisions a North Bay that is working together hand in hand to find sustainable solutions, and we invite you to get environmentally ready with us. We're building a community of learners and problem solvers across all sectors of society by providing enhanced understanding of our connection with the environment and the skill-building experiences that lead towards sustainable solutions. There are many, many ways to get involved with the work that we're doing. You can conduct your own research. You can participate in our Naturalist training programs. You can attend events like these. You can access data. We have a ton of data on our website of all sorts of things that have been found on the preserves that have to do with the cultural and the native, as well as the natural history of the preserves. You could lead or contribute to events such as this if you have something that you would like to share that you think would add to our solutions. Please let us know that. Anyway, it's each of you is a critical element as we work together to address the greatest environmental challenges in history. So today we're going to talk about phenology, a technique for understanding plant life, in this case, and its valuable role in citizen science. Our team today is led by Prahlada Papper, who is an educator, naturalist, and UC graduate researcher. His research at the Hopland Research and Extension Center, which we will be referred to as HREC going forward, involves the genetic and ecological diversity of California's oaks. And he is a citizen scientist fanatic by his own admission. And he supports the phenology team of volunteers that is doing this work up on HREC. Working alongside him today is Hannah Bird, who is the community educator for HREC, and 4 members of the HREC volunteer phenology team. Prahlada will present for about 20 minutes, and then we will have a chance to talk about the data sheet that we sent a week or so again. And then we sent it out again yesterday. Now, some of you may not have had a chance to work with it yet, which is fine. He'll walk us through it and help us understand some of the things it can do and some of the challenges you may run into working with it. If you have questions, and then Hannah's going to say a few words, and she will do an informal interview with the volunteers about their experiences and reactions to the work that they're doing in the field. So if you would all please mute yourselves and turn off your video at this time. If you have questions, feel free to put those into the chat down at the bottom of your screen. I will be monitoring that, and my colleague, Carrie Weniger, will also be helping me with that. So if things are coming in, if there's something like you can't hear us or you've got some kind of technical things, put those in. Even if you have a question that is relevant to the topic that's right under consideration right then, and you don't want to lose it, put that into, and we'll watch it and ask it as it seems appropriate. The team has agreed to stay around for about 15 minutes after the presentation. We will stop at 11, but they will stay around for other questions. And even after that, if you have questions, feel free to email me, and I will put my email into the chat right now. And you also should have my email through all of the correspondence that we've done after your registration. So with that, I thank Prahlad. This is being recorded, and we will let you know probably within the week after the presentation where you can see it. And you're free to share it with anyone you'd like. So Prahlad, I think you are on, ready to go.
Okay, thank you, Margot, and Carrie as well. You guys run these presentations really well. I'm always scared to come on Zoom because it can fall apart so easily, but the ones I've been to that you put on have been really well organized, and I appreciate that. I'm looking forward to this. In a lot of ways, what we're going to be doing today in this next hour is a super condensed version of a workshop that we put on frequently to introduce people to phenology, but that often lasts several hours because we'll go out into the field and take observations together. And so what we tried to do is come up with a way of giving you a lot of those same experiences, but in a sort of a more condensed form. And so I'm going to give a short introduction to phenology, and then hopefully we'll be able to talk about the experience of taking phenology data based on your experiences before today, using the data sheet that we sent out. So let's jump into it. You can see this, I believe. So my name again is Prahlada Papper, and I also like to give credit to Susan Mazer on the first slide. This is a modified version of a presentation that she created to introduce this.
I'll let you do it as soon as I do.
Sure.
Probably Wednesday night.
To introduce the California Phenology Project, which is a sort of a statewide project to look at phenology that we'll be talking about more later as we go on, after I've introduced the general topic. And so this, Margot already kind of went over this, but here's how we're going to break up this hour. I'm going to be introducing phenology here at the beginning, talking about the idea of phenology, which is probably a new term and perhaps a new concept for a lot of people. And then we'll move into discussing the data sheets that we sent out and a little bit more detail about how the Phenology Project works specifically at Hopland REC, and talk to some of the volunteers that have been working with it for a while. So in order to introduce phenology, I think we need to start out with a little bit of terminology clarification, because this is a new term for a lot of people. It's interesting, when I first started giving introductions and talks about phenology 7 years ago, it was always a new term for everybody. It seems like over the past few years, more and more people are hearing about it, which is gratifying for me, I think. But it's definitely still a new term for a lot of people, and so we need to clarify some things. First of all, there's a study of the measurements of people's skulls and what that says about their personality and capacities. That's called phrenology with an R. That's not what we're talking about. There's also the study of it's a branch of linguistics that deals with the sounds that contribute to language. That's called phonology, which is always what Microsoft Word thinks I mean when I write phenology in a document. Instead, what we're going to be talking about is these patterns in plant lives, like the title of today's workshop. It's really a study of recurring life cycle stages and how the timing of those stages relates to the organism's environment. Often people will add the recurring life cycle stages of plants and animals, but I like to make it broader, because it's not just plants and animals that have phenology. All living organisms have these recurring stages in their life cycle that are affected by their environment in some way, and that's what we want to talk about today. So let's look at some examples of phenology and sort of get a sense of what the field is, what the range of life cycle stages that we're talking about is. So obviously things like spring wildflowers are a great example. One of California's favorite phenological events is the wildflower blooms that happen in the spring across a large part of the state. Other common examples, more common especially on the east coast of the US, although we obviously have it here in California too, is the fall color change in the leaves of trees, right? So when deciduous trees begin senescing, dropping their leaves, that's a phenological event. It's an event in the life cycle of those trees. But it's not just plants. So we're going to be focusing on plants today because there's some benefits to monitoring plants. They're just easier to monitor is often one of the main things. But animals have phenology as well. All animals do, really. And one of the classic examples, one of the most well-known examples to most people, is bird migrations, right? So when birds migrate north or south or wherever they're going, that's an event in their life cycle, and it's in some way inspired by the environment that they live in. It's triggered by a change in the environment, and it causes them to initiate this new stage in their life, this migratory stage. And then, like I said, I like to go beyond plants and animals. Always like to broaden our perspective when we think about biology. And so the flushing of mushrooms in the fall, which is a pretty distinctive part, especially here in Northern California. We care a lot about that flushing of mushrooms. It's a big event kind of when it happens. That's a phenology as well. There's organisms, the fungal organisms living underground, and they send up these mushrooms. And that's a stage in their life cycle that's triggered by a change in the environment, by the arrival of moisture and maybe different temperatures. And then viral outbreaks is one I always like to bring up. I brought this up before COVID-19, but maybe COVID-19 gives us a even broader view of it. When a virus reproduces like crazy and creates an epidemic or a pandemic like we're seeing, that's a phenological event for that population of viruses. So yearly there's an influenza epidemic. And that's a phenological event. It's the reproduction of those viruses is tied to the environment. In many cases, we are their environment, and so it's changes in us that cause their populations to boom. And so that's a phenology as well. And so when we're talking about phenology, even though we're mostly talking about phenology in plants, do think about it more broadly. So, like I said, phenology we should think of as a study of recurring life cycle stages, whether they're in plants or animals or fungi or even viruses, which are technically maybe not even considered living things, but they do have a phenology that's tied to the environment. And then we want to introduce just one more term, if phenology is maybe getting comfortable for you. I'll be mentioning several times the idea of phenophases, which is just a specific life cycle stage. So the change in leaf color in fall is a phenophase. When leaves shift from being green to red or yellow or brown, that's a phenophase. It's a shift in a specific life cycle stage.
Prahlada, I don't mean to interrupt you, but there's a sound that we're all hearing, a tapping sound.
You hear a tapping sound?
Yeah, and I don't know where it's coming from. Sounds like typing,
wait.
But not quite.
Do you still hear?
No.
I don't
Yeah,
know what it is.
Maybe it's your are you on your keyboard?
I'm not typing though.
Oh well.
I'm not sure what it is.
I'm not sure either. Okay, but everybody else is muted, so we figured it had to be you. But it's okay. Go ahead. I'm sorry.
Okay, sorry about that. I'm not sure if there's anything I can do about it. Oh, actually, maybe it's bouncing against something. Anyway, all right. Okay, so this is what we want to talk about is these life cycle stages, and specifically we're going to be talking quite a bit about how they're initiated by the environment. But these examples don't end there, and really people are very familiar with phenology. They may not know the term, they may not have thought about it explicitly before, but phenology sort of pervades our life. Once you start thinking about it, once you start seeing phenology, you realize that almost everything in your life is in some way affected by these life cycle stages that happen in organisms around us. And so this is just, I'm not going to talk about every one of these examples in the interest of time, but this is just to give you a sense of how many things that we deal with on a daily daily basis throughout our lives are really, really intimately tied to the timing of these life cycle stages in the organisms we interact with. I will focus on a couple of them though that are especially important. One is wildfires, right? So obviously wildfires have become even more important than they were the first time I ever gave this presentation. Wildfires are really defined by the phenology of the plants that are potentially burning at that time. We talk a lot about fire weather and the red flag warnings that are dependent on that weather. But if it's often a combination of that weather and the timing of the plants. So that, especially in California, our plants tend to dry down during the summer, and they become much more flammable through the summer. And so that phenological change that's happening in California's plants through the summer is a big part of why wildfires are so important in California's ecology. And then the other one I want to focus on a little bit is agriculture. Just because it's such a characteristically human endeavor maybe, and whether you've ever been involved in agriculture or maybe just gardening in your backyard, that's phenology. Agriculture is essentially the same as phenology. Nothing that a farmer does or a gardener in their backyard does is separate from understanding the timing of the life cycle stages of the plants they're growing. So if you've grown tomatoes in your backyard, you've watched the phenology of those tomatoes closely over the weeks and months in order to harvest the tomatoes at the right time, in order to remove pests at the right time, in order to put them in at the right time of the year. All of that is dependent on understanding the phenology of the plants you're growing. So phenology shouldn't be a new concept, even if it's a new term for people. But we can break it down a little bit, and this is what I want to do, is think a little bit more about what is the phenology that we're talking about. And so we can start by saying that one of the ways of thinking about phenology is to really make it clear that plants and animals are dynamic over the seasons. We often like to think of nature as being sort of so stable and unchanging in the absence of human interaction, I guess. But we like to think that nature doesn't change. Nature is incredibly dynamic, always changing, from year to year changing, and from season to season changing. And phenology is really one of the most important ways of understanding that dynamic nature of plants and animals. And so we can divide, especially plants, we often want to divide the dynamism of plants into two distinct categories. One is vegetative phenology, so phenology that affects the leaves and stems and growth of a plant. And then separately, we can also think about the plant's reproductive phenology, which is sort of flowering and fruiting and reproduction. And these things are often happening simultaneously. Leaves and flowers often come out at the same time in the spring, but conceptually we can separate them. And I think that's really beneficial and helpful because one of the goals of phenology is to get a better naturalist's eye. And one of the ways of doing that is sort of categorizing what you see so that it makes sense and you can more easily compare it between one species and another, for example. And animals we can make a similar separation in animals. Animals are really very much, well, plants and animals are both about reproduction almost entirely. Reproduction is the most important thing organisms do. And so again, reproductive phenology in animals is very important. Things like the nesting of birds and estrus in animals or mammals like these dogs. And then maybe some things that are somewhat more non-reproductive, like the timing of velvet on the antlers of a deer, kind of still indirectly related to their reproduction, but we can make these separations in animals as well. But really what we want to think about often when we think about phenology is how the phenologies of different organisms interact with one another. And so classic examples are often plants and animals interacting with each other, although different plants will interact and different animals will also have interactions. But these interactions between plants and animals that are really what ecology is. The study of ecology can often just be thought of as the study of the interactions between organisms. I don't think that I'm exaggerating to say that the majority of those interactions are dependent on the phenology of the organisms. So things like herbivory, shown here with this rabbit eating some leaves, that's a phenological interaction. The leaves of that plant have to be out and tender at a certain time in order for the rabbit to have its food source at a time of year that it needs food in order to fatten up for having babies or something like that, perhaps. Other examples like the interaction between flowers and pollinators, so obviously a phenological interaction. The flowers have to be available at the right time, and the phenology of the insect, in this case a bee, needs to be at the right time for the plant to get pollinated as well. And so the phenology of these two organisms, a poison oak plant in this case and a bee, they have to match. There has to be a match between the timings of these events, the collecting of pollen for the bee and the opening of flowers for the poison oak, in order for the ecological interaction between them to take place. But with all of that change, that dynamism over the seasons, there is a different change that we need to talk about. And this is sort of the crux of phenology now. This is why phenology has become a bigger topic over the past few years. because of climate change, because of global climate change, climate forcing that's going on. And now at this point, there should be no debate over whether climate itself is changing. I don't think there really is anymore in reality. The evidence is clear that climate is warming, climate patterns are changing across every continent and across the globe as a whole. But there's still a big gap in our understanding, even though we understand that, even though we understand these environmental changes, these abiotic changes to the physical environment are happening. There's a really big gap in our understanding of how living things are going to respond to those changes. And this is sort of the push of climate change research over the past decade and into the future, is to understand better how living things are responding to the changing environment. And so this 2007 report for the UN panel on climate change said that phenology, this timing of seasonal activities, is perhaps the simplest process in which to track the changes in the ecology of species in response to climate change. They called out phenology as one of the best ways of understanding how climate change is affecting organisms. And to be clear, if the climate was changing and somehow it could happen without affecting any organisms, we really wouldn't care about that. Climate change is only an important thing because it's going to have effects on plants and animals and other organisms. And that's why we really care about it. And so we really do want to understand well how these changes are affecting us and other organisms on the planet. And phenology is the way to do that because, like I've been saying, the timing of these events, the timing of these life cycle changes is dependent on the environment the organism experiences, the temperature and moisture environment that it experiences. So, beyond the fact that the UN IPCC says it's a good thing to do, why is it worth doing this? Why would you go out and start looking at plants and animals to understand the timing of their life cycles? Well, one is just to understand these effects of climate change and other environmental changes, especially in your local area. So understand these local effects. You yourself have the ability to see with your own eyes the effects on plants and animals, whether they're native or non-native. Second, though, by doing that, and especially doing that in an organized framework like we're going to be talking about today, you contribute to a global understanding across California and around the world. There's researchers collecting this kind of data. And the more people we have collecting it, the broader the network of people with eyes on plants and animals collecting the data, the better our understanding is going to be. It's the kind of data that we need what's called citizen science for. Citizen science is not a new term for most people anymore. It was when I first started talking about citizen science, like I said, 6 or 7 years ago. Now people are fairly familiar with the idea of citizen science. And phenology is one of the best types of citizen science because it's kind of data that individual researchers just can't collect. It takes too many eyes are needed for an individual researcher to really get good phenology data. But by having this distributed broad network of people collecting the data, we get a lot better resolution of what this change is going to look like. And then third, the one I actually really like to emphasize this because I find that it's one of the things that it's what keeps people coming back in my experience, is that by doing this, by going out and making these observations, especially on a regular basis, the improvements to your personal natural history skills and your awareness of the organisms around you and the ecology around you just improves incredibly. I know it did for me. I started monitoring phenology in say 2013, I think. And it made me a better naturalist. It made me a naturalist who sees the world in a different way than I was. I had a naturalist training before that, and I'd learned a lot about natural history. But it wasn't until I really just buckled down and started paying incredibly close attention, which is the only way you can monitor phenology. It wasn't until that I really thought I started to understand how these patterns were interrelated.
Prahlada, it's about, you got about 5 minutes left.
Okay, thank you, Margo. And then the last thing I want to highlight here is that it gives you an opportunity to speak with expertise about something. By making these observations, you gain a type of expertise that no academic research scientist is able to get, because they're so locked into the pattern, the sort of track of research, grant writing and paper writing and meetings and all these other things that take up the time of researchers, they don't have a whole lot of opportunity to go out into the field and really observe things closely. They love to, and they do whenever they can. But by just doing that yourself in your free time, you gain a type of expertise that's incredibly rare in the world today and incredibly necessary. The ability to speak with expertise about the plants and animals and their reactions to changes in the environment is it's an incredibly valuable thing. And the more people that have that expertise and that can speak with that kind of knowledge, the better off we'll be. So in this last 5 minutes that Margot has told me I have, I'm going to just introduce a few different phenology research projects that have happened throughout history so that we can see how useful it can be to have this large network. And the first one I'm going to talk about was initiated by this guy, Joe Caprio from Montana State University, starting in the 1950s. He sent out to eventually about 3,500 people around the United States and into Canada. He sent out clones of lilac, syringa. And so these were all genetically identical clones of one another. And he sent them out across the entire United States and southern Canada and asked people to report back to him on certain events, certain phenological events, when they first had leaves and when they first had flowers, in order to understand how those patterns would change across North America. And his initial goal was mostly these first two goals here, to show the effects of elevation and latitude on sort of the onset of spring phenology, the onset of warm temperatures that initiate leaf and flower growth. And then second, and maybe most importantly, he was interested in creating predictive maps for when it would be safe, what time of year it would be safe to plant crops. And his research was fundamental in creating the USDA's plant hardiness maps. So what time of year can you plant, say, corn in a given location? And these clones of lilacs were really influential, or really able to do that. And it wasn't until quite a few decades later, in the 1990s, really, that people started to realize that this long data set now, that was by that time 40 years old, now 70 years old, has the ability to also show us information about how climate has changed in the United States over that same time. That came a little bit later. And so just briefly, here's the pattern in these lilacs that Joe Caprio sent out and people reported back on. In the spring, they have a simple change as buds start to break. So these closed buds start to break and leaves begin emerging. And this is one of the phenological events, the Phenophases, that the volunteers were reporting back on. And then a little bit after that, they began opening their flowers. And this is also an event that people reported back on. The phenology continues, and those flowers turn into fruits, and the fruits dry at the end of the season. I believe most of the volunteers also reported on those later stages, but it's mostly the leaf out and flowering time that became really influential parts of the data. So here's the map that Joe Caprio was able to create. So every one of the blue dots on this map is a clonal lilac that was sent out to a volunteer, planted in their yard or near their home, and that they reported back data on every year. And you'll notice that it only covers parts of North America, right? So there's actually none in California, or except up in Modoc County, and it's because California actually doesn't get cold enough for this particular clone of lilac to open up distinctly in the spring. And so this particular clone of lilac would not work in California, and it also wouldn't work throughout the Southeast. There's been some other attempts to introduce other species as models like this in those parts of the country, but this lilac project hasn't worked out in California or the Southeast. But what you can see is that based on this, Joe Caprio was able to draw these lines, which are days of the year, the 60th day of the year, the 70th day of the year, the 80th, 90th, 100th day of the year. And those are the days on average when these lilacs would leaf out. So over a 40-year average, or 30-year average, I guess it is here, when did lilacs leaf out at these different locations? And you can see that there's a clear pattern. It's not a surprising pattern for anybody who's familiar with plants or agriculture, that as you're moving further and further north in North America, leaf out date is getting later and later into the year for the most part. But then there's complications going on here in the Rocky Mountains, which is a case where elevation is creating a similar pattern. And things get complicated for sure, but this was really, really useful data for farmers and for gardeners and for landscapers and anybody who deals with plants. And it also became useful for people dealing with animals, because like I said, animals have this phenology as well. But again, this is many individuals averaged over, say, 30 years in this case. The shift in our understanding of this data set came when we started to ask, well, wait a minute, what if we now look at just one individual and track it over time? What kind of information can we get? So let's look at an example of that. And this is from Mark Schwartz. Mark Schwartz was the guy who in the 1990s realized that this data set could be used in this way. It hadn't really been used in this way before then. So here's just a single example, this one lilac that was planted in Essex Junction, Vermont in 1964, 10 years after the project had started, but this person who was monitoring it gave really, really reliable data over time. And so 40 years of tracking that plant, that one lilac clone, this is what was able to be seen, is that the date of first leaf over this 40 years from 1964 to 2004, when this particular plot ends, there's a trend of it happening earlier over time. And the same with the date of first bloom, it's happening earlier over time. Interestingly, though, there's a lot of variation. There's days in the years in the 1960s where the bloom of the first leaf was very early. In 1982, the first leaf was very, very early. And so there's a lot of year-to-year variation as well. But then over time, we can start to see a trend, and we can match that trend to the trend in temperature also in Essex Junction, Vermont. So in the same town, weather station data collecting mean annual temperature, average annual temperature, and see that even though there's quite a lot of variation from year to year, there's also this trend of warming temperature. And the conclusion then that we can draw simply from this, although there's a lot more complex we could do, is that the phenophase onsets in this particular plant have advanced, have gotten earlier as the local climate has warmed.
Great. On that note, Prahlada, could we like to have Hannah kind of step in and talk with our volunteers, tell us a little bit about what's going on at HREC, and bring our volunteer team in. Thanks, Prahlada. That's a wonderful introduction, and I think it gave everybody a great deal of information. And if you, Hannah, you've unmuted yourself, I see already.
Can I check, Margot? Everybody can hear me okay?
Yes, we can.
Great.
You may want to go into, I think actually still stay in speaker view, I think works for most people.
Okay.
And if you want to go ahead, and then when you're ready to do your interview, your volunteers can unmute themselves.
Yeah, that's wonderful. So Prahlad, thank you so much. I always get so much out of hearing Prahlad speaking about phenology, because I feel like it connects us to the bigger reasons why we're doing this. But I think what's wonderful, and maybe what I'm bringing now is, dare I say, I'm bringing the selfish perspective of what hooks you in and keeps you coming back as a volunteer. So just to start expressing that, I wanted to explain the site I work at. It's part of the University of California Agriculture and Natural Resources Division. It's a site which is about 5,358 acres, and it's a research and extension center right in the southern part of Mendocino County, so in Hopland. It's a beautiful site, and I feel I have to just share my screen. Oh, okay. I don't need to share my screen. It says I'm not allowed to, so that's fine.
Well, you can. I can give you the right.
Okay, that would be great, Margot. I always feel like it helps just to have a picture of our site, so if that's possible, that would be lovely. And so on this site, which has been owned by the University of California now for nearly 70 years,
you go.
Thank you so much. There's been various different research studies conducted. Thanks, Margot. And also, importantly, the part that I play here is the education side of things. So we first started doing phenology on the site about 5 years ago now, when Prahlad was involved in studies here, and we got talking, and he conducted a volunteer training to allow people to get connected to it. And I have to admit that I think the motivation for most of the folks who came along to that was that this was one of the few ways that you could gain access to the site fairly frequently and enjoy a nice hike. And so they were like, all right, if that's what it takes to come and look at some plants while we're out there enjoying being outside, that's fine by us. And Prahlad did his training, and we started with this small group of people. And, you know, this is what's just amazed me, is it's obviously something that gets you utterly hooked, because they keep coming back. So some of our volunteers now have been coming for pretty much that entire time, that whole period of about 5 years. And I think now it's changed from just an opportunity for a nice hike, that's still in there as well, but to something which is about the connectedness to the species that we're monitoring. So we have 30 different plants on the site that we're monitoring. They're in 3 different locations. I'm just going to scooch ahead a bit. So just to show you, we do all sorts of different things here, including education. Here is one of our awesome volunteers, Woody, out looking closely at one of the oaks on site. So 30 different plants in 3 different locations. And just to be clear about this, this is not just all fun and games. A phenology study here, day, will take at least 3 hours. And my volunteers have been coming out in all kinds of weather. Even when it's smoky, we always try and figure out, okay, can we handle it today? Is it going to be safe for everybody to be coming out? Not only that, but we also had a fire come through the site, and the river fire burned through the site in 2018 and burned about 2/3 of the site, which meant that 2 of our phenology research areas were burned. And it left us kind of just for a little while not knowing what to do. And again, really out of the enthusiasm of the volunteers, we then decided to connect with Pepperwood Preserve, who had had a similar situation and had continued to collect data on those same plants, the ones that they could do, the ones that were not totally killed, to see if they were going to come back after the fire. And so we added in this other layer, which is slightly separate to the things that Prahlad has talked about, but was fascinating in our understanding of how native species in California are adapted to fire. And personally, I felt like it was incredibly hopeful to see what had looked like blackened, charred, dead plants coming back again. And, you know, I wish I could just regrow from my base when I was feeling kind of old and tired, but that's what we saw them doing. It was fabulous. So I'm just going to stop sharing that now. And I'm going to pass over, I'm going to be checking in with my volunteers with, we have Janet McLeod, Ingrid Johnson, Woody Hudson, and I think we have Eileen Weeks, but I believe she had some dental treatment, so I know she's not going to join much of the discussion. But I thought it would be a fun time just to understand, so what is it that keeps you guys coming back again and again for years, through COVID, through wildfire, through smoke, through all of these things? So I'm going to just start with just a couple of questions. And Janet, maybe I could just start off with you. So what is it that keeps you coming back?
Unmute yourself, Janet.
Thanks, Margot.
Thank you.
There you go.
Hi. Yes, it's definitely the plants, but it's definitely the people too, because, you know, we've developed a relationship with our team members, and the plants really do become like old friends because you have so much history with them. So I think that's definitely a real draw. And also it is the ranch. It is so beautiful, and to get to go to those 3 sites, like Hannah said, because it's 5 years ago when it started, it wasn't open to the public very much. And we'd lived in this county and, you know, heard about this place for so many years. And so to all of a sudden have access and drive our own cars up to these amazing views and everything like that, it was pretty special. So that was definitely
Thanks, Janet.
keeps
That's
me
great.
coming back.
Woody, what about you? What is it that keeps you coming back? Anything to add to Janet's?
I think you and Janet pretty much covered it. But one other thing that keeps me coming back is all the other interesting things that go on down at HREC. There's other researchers doing other different projects. I've been involved in helping with researchers doing studies of deer, collecting scat samples and trying to figure out through DNA analysis how many deer are actually on the property, and also setting traps to trap the deer so they can have collars mounted on them to monitor them. And there's various other interesting research projects that have been fun to kind of be marginally part of, in addition to doing the phenology. I got started in the phenology about 4 years ago after the program had been started, and was really interested to learn more about the phenophases that plants go through, other than just appreciating looking at them now and then. And it's then when the fire came through, it was more compelling to keep coming back to see how the plants survived, or which ones did and which ones didn't. We started monitoring some manzanitas after the fire to see how they would come back, and they didn't. They were totally cooked. And it varies tremendously too. There's a huge big oak tree that was killed because it had a really strong stand of grass around the trunk, and it totally burned the cambium, and the tree tried to come back. It sent out a lot of epicormics resprouting up in the branches, but that didn't last long, and now it's gone, or it's dead. It's not gone yet.
And it's taken the monitoring for about 2 years for us to see that process, which is.
And then there were other small plants. There were some sticky monkey flowers that survived just because they happened to be in a place that the fire kind of skipped over. It's interesting to see where the fire burned and where it didn't, and how badly it affected a lot of the plants that we were monitoring. About 2/3 of the plants that we were monitoring were in the burned area. So it's been interesting to see how all that happens. It's kind of like a continuing story. You have to keep coming back to see the next chapter.
I love that, Woody. That's great. Ingrid, what would you like to add?
I think it's been said. It's been said. It's the love of the area. It's the plants, knowing them even better. The people that I work with, because I'm learning constantly from the people I work with. And I also must say, Hannah, that it is your welcoming spirit. You have been real welcoming glue to all of us, and it's been felt. And
I'll pay you for that later. Thank you, Ingrid. That's great. So Ingrid, maybe I'm going to start with you this time, if it's okay. What has surprised you as you've been doing phenology observations?
Surprise. What has surprised me is the extensions that I've made in myself. I am a pretty casual person. I don't like to take notes. I don't like to pay a lot of attention to things. I don't like to even read explanations much. I like to just sort of find out how it goes. And I really learned that about myself. And I so appreciate working with other people that are much more careful than I, and have directed me in areas that I can really contribute to, because I know how to be more careful. And I can't even remember what you asked, but anyway,
Okay,
that's one of the points of.
Yeah, no, that's great. We were thinking about what surprised us, and I think that's a really good point, that it takes a team with different skills to be able to work through some of the challenges of making an observation. Janet, would you like to add anything about what surprised you?
I think one of the slides that Prahlad had was about, his deepening observation, and I really find that that's true. And I think as all of us get so delighted over seeing, like, for example, this year, this beautiful old bay tree we've been monitoring, and another one right next to it is gone from the fire. And this year, I think I've been doing it about 4 years, we've seen maybe 1 or 2 bay nets during this time of year in the past, and this year the tree has over 1,000, and it's so fun to just look up and see that there they are. And other times like that, we've looked and looked years with no acorns, and then there'll be a year where there's a big amount of acorns that we find, or even a small amount we get excited about. But just, I think that idea of seeing more deeply And I think another thing that is kind of a surprise too is just feeling like I'm doing one little more thing to contribute to climate change and to science research, because I think we all, I try to do things in my own life to just kind of reduce the carbon, footprint. But this just seems like, oh, there's one more thing this fun thing to do, and it's also contributing in that way. So that's been good.
And I hope we'll get just a few minutes at the end of this just to return to Prahlad to recognize how folks can all get involved. I certainly have my own hopes that some folks who joined this will join our phenology team. We'd love some more phenologists.
Yes.
So as we get to the end, I'll share my email address in the chat if anybody's feeling that this is a team you'd like to be on. It definitely is. They're awesome. Woody, is there anything that's been surprising you as we've gone through the phenology process?
I guess for me, I don't have any kind of biological or botanical background at all. So just finding out how much more there is to the plant's life cycle and what I had to just observe by living in an area where there's lots of trees, that was surprising to me, that there was so much more going on there than I'd ever thought about.
I think that's a really good point as well, that don't feel that you have to be an expert botanist. I am absolutely not. I will admit that my first phenological observations were kind of ruined by the fact that I'd been taking them for a while with a friend, and then Prahlad told us that the species we were looking at was actually something completely different. So, but still, it doesn't matter, right? You can get hooked, and you do look more closely, and that was a learning experience too. So I think it has to be acknowledged that not all phenology observation sessions are just fun and laughter. There is some rather tricky times as well. And so my last question to the fabulous citizen science volunteers is, what is challenging about coming out? And you can be honest, we're all friends here. Woody, I'm going to ask you first, if that's okay. What is challenging about taking these phenology observations?
I think as you get into it, start trying to figure out how to answer some of those questions on the data sheet, and find out that a lot of it is kind of subjective in how you come up with an answer, like how many leaves are on the tree, or whether I'm trying to find acorns on a big oak tree where you can't get anywhere near the top of the tree to see what's going on up there. Those are kind of challenges. It's some challenging to find out exactly how Nature's Notebook thinks we should be answering those questions, and come up with an answer that all of us can agree on.
Thanks, Woody. I think that's definitely the case. Janet, what have you found challenging?
I think the data has been a little challenging over periods because it's changed over time. And after the fire in 2018, Nature's Notebook didn't have a setup for fire damaged plants. And so we came up with quite an elaborate spreadsheet, and it really added like a couple more hours. It was just sometimes the data is quite tedious, and we couldn't take the data in the field for the first years really. And then we'd have to come back and use kind of, old equipment to try to get everything on there. So now, lately, since COVID, that is one good thing. We have been able to put the data mostly in the field. And that has really helped, because for me, I think the field is the most fun and the data is the most tedious. And so, but as we've added new species, so now we can take most of it in the field, but some of it still relies on paper because things haven't been put on Nature's Notebook yet. So at some point it's going to take some time to put all of that data on. So I think a lot of issues around data have been a challenge for me.
I don't think many folks choose data entry as their favorite part of phenology, but I do have to say we have to give a appreciation to some awesome volunteers we've had as part of the team. Susan Schmill and Margaret were involved in hours of data entry, and Woody as well has done definitely his fair share of just straight data entry. So it takes some of that effort as well.
And we have a whole year's worth of data that is not on Nature's Notebook, with photos that Woody has really organized with sequential, sprouting out and changes. And so it's pretty amazing. And, hopefully at some point, Nature's Notebook will have a part that will just be related to fire damaged areas too, that can be used.
Photos that Janet took, which she did a wonderful job of.
Yeah. So I know we're moving to our last few minutes, and Ingrid, I know you will be honest with me about what was challenging for you.
The disappointment of the fire. When the fire first happened, I thought, what's left to look at? I mean, and then coming to the understanding there's a lot left to look at, because things continue to change and to grow, and it's been such an interesting part of my being there. So the difficulty turned into a wonderful experience, and being with Woody and Janet and Eileen, finding out their strengths. Janet and Woody were the expert picture taking team. Eileen always had the binder to tell us more, give us more explanation. So it's been wonderful. The team, and the area.
Yeah, absolutely. So Margot, I know we're down to our last 7 minutes now. I'm wondering whether we have time just to check in with Prahlad, because I know we haven't been able to just check back in on the time that people have spent looking at their own observations. Is that possible, or do you want to do it?
It is. I'm trying to think of the sequencing of this. I have just a few words that I need to say. And I want to give, maybe I'll do that now. And Prahlad, then I will let you take off, and people can leave when they need to. And we can go as long as you have time to discuss with us. But let me do my outgoing comments, if I can find them, and we can go from there. So most, I want to thank you all so very much for sharing your personal experiences as well as your knowledge and your skills. And I hope you all learned a lot, and there's still a lot more to be learned. I think as we go on, I'm particularly interested in what Prahlad has to say, because I found that data sheet really hard to work with. So I'm interested to know how to make that easier. What we participated in today is the 10th of about 20 events that the center has planned through December. And you can find out the rest of them on our website, which is cei.sonoma.edu/calendar. And feel free to spread the word to anybody and anywhere, since we are remote. There are a few programs coming up in the future that I want to call out. One is going to be on ticks, which is also going to be focused on HREC. And the researcher, Emily Pascoe, who's a UC Davis researcher, will be talking about the work that she's been doing up there that is also somewhat fire related, but it has to do with the hosts and the predators of these small but dangerous little creatures. And then in December, we will have an unusual photography program on assistive technology that can help people get into the field and enjoy the world of taking photographs. We also have a whole series where there are 2 parts left to this series called Live from the Field. This is really unusual. It's an unusual series because it involves biological field stations from all over the world. And the format is there are researchers from 3 different stations talking about a topic. The one we did most recently was on harmful algal blooms. And they talk about the research that they're doing and a little bit about how they got into the field. The program is geared toward undergraduates, but certainly I am not an undergraduate, and I found it fascinating. So it's open to all of us, but very, very interesting. And Carrie is putting up some things in here about these programs that I'm talking about now, which can give you some links and some information on that. We have a whole series coming up, but those I particularly wanted to point out to you. So thank you all so very much, and I would like to have Prahlad now go back over that data sheet and spend as much time as he is able, and understand that people need to leave whenever they need to leave. And again, thank you all to your volunteers for all of your work too. I want to get something like this going up in the gallery, Carrie. We'll talk about it. Okay, you're back on, Prahlada.
Thanks, Margot, and thanks to Woody and Ingrid and Janet too, because sort of they went over with the things that I would have wanted to talk about with this data sheet, which is what's great about doing it, but then also like what's so hard about doing it.
Can you put it up on your screen?
What's that?
Can you put the data sheet up?
Yeah. So this is the first time I've ever asked people to just go out and collect phenology data without explaining what phenology is to them or having any time in the field with them to walk through some of these questions. These are tough questions. Actually, the first thing I wanted to do was for people who did have the chance to take data, use this data sheet, I'd like you just to throw into the chat what species you monitored. If you know what species it was, and maybe you didn't, that's fine too. If you know what species you monitored, just put that into the chat, because I'd be curious, I'd be interested to see sort of what the variation was. I told you guys you could just look at whatever plant you wanted to, one or more. So I'd just be curious to see what people were able to look at. And then what we want to do for this last few minutes is talk about the experience of it and get feedback from you about what was difficult, what you found interesting, what you needed more explanation on, because we know that there's more explanation needed, and that's one of the main advantages to doing this in an organized group, like up at Hopland or at Pepperwood Preserve, or they're really all over. And I guess the thing, I didn't get to talk about it in my presentation, but I should mention now, if people didn't go to the website, that this data sheet comes from a website called Nature's Notebook, which is an organized attempt to get citizen scientists out collecting phenology data. And they've created these protocols, they've created this set of questions and definitions, and they've also created an online database where once you've collected the data, you can then enter it and it becomes accessible to the public and accessible to researchers really easily. And so one of the reasons why I had everybody go out and do this is because I hope that you will get over the initial difficulties and want to continue doing it either on your own, and you can do it in your backyard. Lots of Nature's Notebook entries come from people's backyards, or you can get involved at somewhere like Hopland and continue doing it. So actually, I think what we can do now is, so people can definitely unmute themselves, and if anybody has questions, I'd say pipe up and ask them, but I might want to also just maybe go through the data sheet and point out some of the difficulties. But
can you zoom in? Can you zoom in or at all on any of it?
Yeah, I should be able to zoom in.
Because we really can't read it.
But actually, so yeah, first I just want to make a call out. If anybody has like a question or a comment about what this experience was like of collecting this data, I'd be really interested in hearing that. And then maybe we can quickly go through some of the questions and talk about both why they're useful as well as why they need to be so damned complicated.
So can you all unmute yourself and jump in?
It looks like that's cool. A lot of people were looking at oaks. I know, Margot, you looked at an oak. Yeah, so I guess one thing I'll say is that we talked, so the volunteers and I and Hannah talked a little bit before sending out the data sheet about whether to send out the full data sheet with the full set of questions, which are very complicated. And what we decided was that yes, we should. So I created a simplified data sheet that might have been a lot easier for people to collect data using. But we decided not to send that. And I think the best reason for not sending it was that it's the difficulty of these questions that forces you to look really closely. And when I have in the past made simplified data sheets and shared those with people, it becomes really easy just to check yes and no and move on. But it's that act of trying to estimate what the size of these leaves are compared to their theoretical maximum size. The difficulty of that is what makes you look really, really closely, which is the real value of this. And in the process of looking really closely, you may notice something really simple that you would have missed otherwise. You might see the very first flower bud that otherwise you would have missed if you hadn't been looking so closely. And so it's that there's this balance between how difficult some of these questions are and how technical they become, and the value, the benefit that you get out of it, the benefit in data and also the benefit personally, because like we've been saying, a lot of the benefit that we get out of this is greater natural history understanding. And I really like what Janet and Ingrid said about the personal connection that you get to the plants that you're monitoring. I think that's really undervalued when we talk about the value of citizen science. We talk about the data that's generated, and we talk about the engagement of the public, but we don't talk about this like personal emotional connection that it can help build between people and the nature around them. And it's struggling with those really difficult questions, I think, that helps build that.
Prahlad, can I just butt in for a second? So one thing that I so enjoy when you've done trainings with us in the field, so I think one thing that's awesome is that you can do this in your own backyard, and you can find a species that's of interest on Nature's Notebook, and you can be recording that. But there's something wonderful about doing it as a team as well, wonderful and challenging, because when you're trying to come up with a decision about an observation, not everybody agrees, always. And so I've seen Prahlad, when he's training folks up, say, do you know what, you're just going to have to do rock, paper, scissors for this. You know, there is a point at which we could spend 3 hours debating this, whether there's hundreds or thousands of seeds, or we could just say, do you know, this is a difficult enough decision that we're just going to let fate decide. Right, Prahlad?
Yeah, I mean, and part of that is just that when you're, so a lot of the difficulty, well, let's say most of the difficulty in this comes in these follow-up questions. When you're asked, do you see breaking leaf buds? And then you're asked, if you said yes, you're asked, how many did you see? And that's where a lot of the difficulty comes in. How do you count 10,000 breaking leaf buds, for example? And a lot of the difficulty really actually ends up being on deciding between two adjacent categories. People get really hung up on, is it 11 to 100, or is it more than 100? And one of the ways of thinking about scientific data collection is just that if you're really trying to decide between two adjacent categories, you should kind of pick one of them randomly, and then pick one randomly the next time. And over time, that will average out. It's difficult for people to get comfortable with not being really exact in data collection, but it's actually a really important lesson for science, at least, and probably for general life, to not get so hung up on the exact answer, and to sort of really think about the bigger picture about why you're collecting this data. Yeah. So I guess, so I don't know, did people not have any issues? We assumed people would have a lot of difficulty with these questions and would get pretty frustrated with them, because that's been our experience, is that it can be really frustrating. And it takes a long time. One of the things that you'll notice on this, so I guess I can say that for that example I gave of the increasing leaf size and the percent of full size, what percent of full size are these increasing leaves? And the definition of what that means kind of requires you to have been monitoring the plant for quite a while. It assumes that you know what the full size is eventually going to be. And it's been one of our experiences with phenology monitoring that almost the entire first year of data is training. It gets entered to Nature's Notebook, and it's very useful, but people who are taking the data often don't feel comfortable making these estimates until they've watched the plants for an entire year. And so really, if you do go into taking this kind of data, you should consider that your first year is going to be a training year. So don't expect to learn this in a month, because after 1 month, you'll be looking at an entirely new set of phenophases that you didn't look at the previous month. And it's not until you've come back around to the same phenophases again that you will be able to have experience with them, really.
Ned, I see you there, and you did the stinkwort and the panicled willow herb. Do you want to talk about your experience?
Well, sure. It was, I mean, the reason, I'm no expert, but I have eye naturalists, so it helped me identify these, and they're right in our yard, so I've been watching them and not cutting them, because they're actually very interesting plants to watch develop, especially the willow herb. But it just, what we were just, so my experiences of it was, I was trying to put something down, and handily there's a lot of little question marks, so I was able to circle those if I wasn't sure. But that led me to wonder about how the data is used later. So when you're talking about maybe the first year of data collection for this, and maybe for a lot of other citizen science stuff, being mostly training, so is that like another data point in there that they know the person who collected it is still something of a novice, and so a researcher can look at this and say, okay, this data point might not be as reliable somehow. Is the data set big enough that you can actually do that sort of stuff? I mean, where is, I just don't know anything about Nature's Notebook, you know. But in terms of using the data sheet, it actually helped, the more details helped, I think, like really look at the plant and figure out what the heck is going on here. And I could see that that would need a lot more familiarity, you know, to make it work out. But it was fun, definitely a fun exercise. And the plants that I chose aren't like big and robust things like an oak tree or something like that. So is this sort of data collection appropriate for, like, you know, little annuals or things that die away really fast? Are there different sheets you would use for a different type of plant, you know?
Yeah, so if you, and I encourage everybody obviously to go to Nature's Notebook and look up some of the variations that there's over 1,000 plant species in their database, which means there's lots of plant species that are not in their database, although they're always adding new ones. But yeah, it includes a lot of small herbaceous plants, and more or less these same questions are asked, but there is some variation. So I've just listed like 2 broad categories here, evergreen and deciduous, but there's actually quite a bit more detail that can be gone into, and some of these questions do get dropped out in certain cases. So even for certain deciduous plants, you won't ask every single one of these questions if it's not totally appropriate. But for the most part, it's the same questions. So I don't, an herbaceous plant, you can ask almost all of these same questions. You just have to interpret them a little bit differently sometimes. And that's what so the information on Nature's Notebook can be really helpful with that, because they'll give you specifics. So they'll say, you know, for California buckwheat, think about this when you think about leaves, for example.
Okay, so this is a more generic type of data.
This was sent out to be very generic and doesn't have any of that detail. And actually, so Nature's Notebook will also have photo guides for a lot of species, not for every one of their 1,000 species. Plus they have several hundred animals. So there's also animal phenology data on Nature's Notebook. And so they won't have photo guides for every single one of the species that they have in their database, but for a lot of them, for the more commonly observed ones, there's actually a photo guide, so you can see what you're supposed to be looking for. And that's a huge part of it. That's a huge part of that first year learning curve, is learning what the flower buds of California Bay look like. If you haven't looked closely, you probably don't know. You've lived with California Bay all your life and may never have known what the flower buds were. It takes a while to figure that out, unless having a photo guide can help, but it just really takes the experience of doing it. And then actually I wanted to address your other question too, which was about, is that sort of learning curve taken into account when somebody goes to analyze this data? And the answer is pretty complicated. I think, to be honest, the answer is that researchers who go to use this database are going to assume that it's all collected in the learning curve period. Just to be safe, they're going to assume that it's all collected with a certain amount of inaccuracy and uncertainty. And so that's the way it's probably most commonly taken into account. And what that means is that some of these questions end up getting ignored, because the researcher just assumes that the data is going to be too messy. But then you did bring up the point that once the data set gets big enough, a lot of that sloppiness kind of washes out statistically. So when you get into the statistics of analyzing a data set, the larger it is, the more inaccuracy becomes just noise, and there's still a signal that can be noticed in that noise. And so that's one of the goals, is to get this data set big enough, even if there is quite a bit of uncertainty in the way people are collecting it. But then the second part of it is, I mentioned at the very beginning of my presentation, this California Phenology project, which the way to think of the California Phenology project maybe is like it's the elite team of phenology monitoring. It's a team of phenology monitors across California that have more detailed training and more rigorous protocols. And so if we know which sites are California Phenology project sites, like Hopland REC is a phenology project site, then we can have greater confidence, because we know that all of those volunteers have been trained in the same way and are using the same data sets and the same rigorous protocols. So that's another way of addressing that, is to have subsets of the data that are more detailed.
How big is the data set you've got now, or is it sort of a collection of a whole bunch of things, or is it still growing?
The Nature's Notebook data set is growing very much. So Nature's Notebook is part of a national project, the USA National Phenology Network. And I haven't even been keeping track of their statistics over the past couple of years, but I know that their data set was growing exponentially every year or so. It was doubling. The number of observations was doubling every year for the first 5 or 6 years. That probably still is, would be my guess, because citizen science is getting more recognition, and phenology is getting more recognition, and this desire to understand your local environment. And so, yes, the Nature's Notebook data set is growing, and I know that the number of plants and animals that are included in their database has grown. Every time I give a presentation like this, I have to update it, because it goes from 200 plant species the first time, and now it's up above 1,000, because they keep adding more. If somebody wants to request a plant that's not in their database, they'll usually just add it pretty quickly, and so that has been growing as well. Yeah, it's definitely a growing database, and it's been used for quite a bit. Quite a bit of research has come out using this citizen science network. There's people in academia who study citizen science always whine about how little of it actually ends up getting used in published research. So there's definitely more that could be used, but quite a bit of really good papers have come out looking at both national, sort of North American trends, as well as species-specific trends. So like Margot said, I study oaks in California, and 2 or 3 papers have come out looking at specific patterns in the timing of leaf out and flowering in oaks that have been based on California Phenology Project data. So once the data...
There are quite a few people who are still on who haven't said anything, and I'm wondering if any of you had an opportunity to play with this data sheet, and if you have any thoughts or questions for Prahlada or the volunteers. I think we still have our volunteers left. Hannah has had to leave, but we have other people here, and if I don't hear any, and there were no other questions except for my question, which was sort of a general question I put in there. So when I guess it was Janet was talking about the bayberries, and I noticed that on some trees too. And where do you go to find out the why? I mean, for people who aren't biologists, I want to know why this tree this year is just dumping everywhere, and last year there was nothing. Where do you go to find out those whys, those answers?
So I was at Kate Marienchild's talk last time, and there's that similar why question about masting years in oaks. I think she said she wasn't going to address it. That's what it was. Somebody asked, and she said, "I don't really know, so I'm not going to address it." And
that was helpful.
It's a very difficult question to understand why, right? So this database, the California Phenology Project database and the Nature's Notebook database, is really just about seeing the patterns.
Right.
Explanation of why these patterns exist is definitely an additional level of research. And well, I mean, we can start out simply by saying it's something in the environment, right? So something in the environment is causing, California bays in this case, to produce a lot of fruit in a certain year, a lot of nuts. And people have a lot of hypotheses about what it is. We can talk about what some of those hypotheses are. So
right.
One hypothesis is that it's a coordination of different climate factors, right? So the right temperature, the right moisture coming together at the right time of the year is one hypothesis. That's called the resource abundance hypothesis. That it's just the right resources abundant this year, and the plants respond to that. But then a whole second hypothesis is that it's called the predator satiation hypothesis. And the idea is that these plants, like an oak or a California bay, produces a mass of fruits called a masting year, produces a ton of fruits in a year in order to overcome the predators of those fruits. So that even though maybe the majority of the fruits will get eaten by predators, the fact that there's so many of them in a certain year means that that's the year that they will be able to establish seedlings. And in most years, they may not ever establish seedlings, because if they don't produce 10,000 fruits per tree, then predators will eat all of them. And it's only in the years where they can produce a massive number of fruits that they're able to overcome that sort of predator pressure. So that's these two different hypotheses, and they're probably not contradictory. It's probably a combination of both of those.
Yeah, interesting. One person put in the chat, and I thank you for this, that she felt too intimidated to try to use the database. I felt a little bit like that too.
So let me just say that the vast majority of people who are using Nature's Notebook are just ordinary people who have had no special training except reading what they found on the website and going out in their yard and doing it. There are, like I said, there's these elite teams like the California Phonology Project and some other projects around the country that give more training, but that's a small subset of what's on Nature's Notebook, and the vast majority of people who are entering data on Nature's Notebook haven't had any special training. So you shouldn't feel, well, I mean, you should feel intimidated in some ways because that's going to make you sort of better and paying more attention. Don't be sloppy, but you shouldn't feel like there's something strange about doing this without having a lot of extra training. The vast majority of people who do it don't have any special training.
Okay.
And I would definitely encourage people, if you enjoyed this or you think that with enough time you would begin to enjoy this, start doing it. Keep watching whatever plant you picked out this past week, go out and look at it again this week and look at it again next week, and start doing it more and more often, and create a site on Nature's Notebook and start entering your data online. You shouldn't feel like you're not allowed to do that, because that's entirely the point of Nature's Notebook, is for ordinary people who have had no real training to start entering this data, and you'll get better and better at it over time. There's quite a lot of information on Nature's Notebook as well. So if there's specific questions that you found really difficult in this, there's a whole frequently asked questions page that goes into quite a bit more detail. And I'm always happy to answer questions as well. You can contact me, or you can contact the volunteers at Hopland who have become real experts in this, and they're willing to help as well.
I found it fascinating. I found it fascinating, Margaret. That same person typed in the chat that now she feels a little more confident, knowing you can enter your best guess and will learn over time. So
that's what we're all about, right? Is to take people from that sense of powerless to being able to think, well, now I can try it, which is great.
And Ned pointed out that there's lots of question marks on there as well. And so when you're really, really unsure, don't forget that I don't know is the right answer when you don't know.
Yeah.
You can also leave something blank and not enter anything at all. That's also a choice, but might as well enter question mark.
Well, I think doing it within Nature's Notebook would be helpful too, so you can have some other resources to go back to. Well, thank you, Prahlad, so much, and to Janet and Ingrid and Woody and all of you guys for helping. I know Hannah had to leave, but thank you for the presentation. Very interesting, and I hope we'll all go out and take our new skills and track our plant activities. And we'll hope to see you all soon. We have another event coming up on Thursday on tracking, animal tracking. Hope to see you guys there. Thank you.